Okay. I want to start with the acknowledgement of the country. I celebrate and acknowledge the traditional owners and custodians of the land on which we meet today, the Honorary people of the Kulin Nation. I pay my respects to their elders past and present and embrace their continued connections to this place. I'm Bernard. I will host this seminar today, and thank you all for joining. It's my great pleasure today to introduce today's speaker, David Vaux, or Davo, as he's better known at WEHI, I think. Davo spent his extensive scientific career investigating programmed cell death, or apoptosis. He initially graduated in medicine from the University of Melbourne in 1984, followed by a PhD at WEHI in 1989. After a postdoc in Irv Weissman's lab at Stanford, he returned to WEHI in 1992 and has stayed at the Institute for most of his career since then, until his retirement at the end of 2019, I think, right? He's still quite active in his retirement, and one of his big passions outside apoptosis is science integrity and ethics, and that's what brings him here today. He was WEHI's deputy director on science integrity and ethics, and is still a strong advocate for this. So just 4 weeks ago, he published an article for The Conversation to outline why Australia needs an office for research integrity. So I urge you to have a look at that. And his presentation on experimental design and statistics, he will present this today, or as he calls it, research integrity, the good, the bad, and the ugly. Thanks, David.
Thank you for the very kind introduction, and the first mistake is clapping before you've heard my talk. I'll accept applause afterwards, or boos, or whatever. Now, this talk is the tip of the iceberg. I've got lots of stories I could tell you, but it would take far too long. So I'm going to concentrate on two things. I'm going to concentrate on a little bit of statistics for people who aren't statisticians, things to look for in papers as you're reading them, things to do when you're in the lab, and I'm going to give one case history, just one, about a researcher who wasn't doing the right thing, but show you the story of how that came about. I put in new batteries in my laser, and you can see it's a very powerful one. And I notice that all of you are towards the back of the room. So I'll be asking you questions as I go along. And if you feel your retina burning, that's because I'm pointing at you. So this is a paper published over 10 years ago now, why most published research findings are false. Every year in PubMed, there's another 1 million publications. And John Eney's mathematically proved that the conclusions of most of them are untrue. So it's impossible to read 1 million papers every year. So this talk, hopefully, will give you some hints and tips as to how to figure out which papers you should read and which ones you should throw immediately in the trash. Now, a lot of people thought that John Ioannidis was exaggerating, and there's no, this is a theoretical basis. He was mainly criticising use of p-values, but there is some evidence to support the fact that, he was onto something. And this is a review published by Glenn Begley. Glenn Begley did his PhD here at WEHI, also worked at Royal Melbourne Hospital. But he became the head of oncology at Amgen, one of the world's biggest biotechs. So his job was to find new drugs to treat cancers and new targets to hit with new cancer drugs. And the way at Amgen they would find these new molecules is by reading the academic literature. But they found that often, if they'd see some new molecule, described in Science or New England Medical Journal, when they tried to test it in the lab, they couldn't reproduce the findings. So in frustration, he published this commentary and published it in Nature, where they looked at 53 landmark studies. So these are preclinical cancer studies published in the best journals in the world from the best institutions in the world. And for those 53 papers, they tried to reproduce the findings, the key findings in those papers. And if they couldn't reproduce them, they contacted the authors of the papers and said, look, we'll keep your names anonymous, we won't reveal who you are, but can you help us reproduce your published findings, or can you reproduce them again? We'll give you all of the resources necessary, but can you reproduce them while we watch? And they found that even with the help of the authors, they could only reproduce the findings in 6 of those 53 papers, so about 11%. So Glenn concluded that 90% of papers published in preclinical cancer research, and these are landmark papers, the best, couldn't be reproduced. So once again, so 1 million papers every year, probably 90% of them can't be reproduced. So how do you figure out which are the good ones and which are the ones you should throw in the trash? I came to similar conclusions about the poor quality of publications partly from reading this one edition of Nature published in 2004, and the next examples are going to all come from this one edition of Nature. So Nature, of course, publishes papers in biology, in physics and chemistry, all of the sciences, and Nature and journals don't just publish the conclusions in the title and the abstract, they're meant to contain the evidence that supports those conclusions. So one tip is if you want to understand a paper, you can't just read the abstract and the title, you've got to look at the data, look at the materials and methods. So here's a typical figure in a paper in Nature, and there's a figure legend, and here's some Western blots detecting protein. Here are some graphs. In these bar graphs, you see these little T-shaped things in the bar graphs, and I'll ask the person in the front right there, what do these little T things represent? And I'm looking for two words, one starting with letter E and one starting with letter B. Error bars, very good, very good. So we're on to a great start there. Next person along, can you shout out, what are the error bars in this figure? That's a hint down there. Standard deviations, very good. Right, so here's a graph, means plus standard deviations. Here's another graph, same paper, same edition of Nature, different paper, same edition of Nature. Once again, there's graphs, and I'll now ask the person with the red top on the edge there, what are these T-shaped things called? Error bars. And the next person along, this is test your eyesight. What are the error bars in this case? The guy with the black top. Just read it out.
Mean standard error.
Yeah, SEM, which stands for standard error of the mean, right? So that's what the error bar's there. Here's another figure, same edition of Nature, but a different paper. Now I'll pick on the guy with a stripy shirt. What are these T-shaped things going up there and down there, and up there and down there? What are the T-shaped things called? The general error bars. And the next person along with the white mask, what are the error bars in this case? Confidence intervals, very good, very good. So excellent eyesight as well as correct answers. Here's another figure, same edition of Nature, different paper. The guy in the middle there, right in the middle with the beard, yeah, you. What are these T-shaped things called? Error bars. And the lady in the yellow top, what are the error bars in this case? Doesn't say. It's a trick question. Doesn't say here in the figure legends or in the materials and methods. So, we don't know what they are. And here's another paper, same edition of Nature, another figure, error bars, doesn't say what the error bars are in the figure legends or materials and methods. Here's another figure, same edition of Nature, error bars, doesn't say in them. So it turned out in this one edition of Nature, there were 10 papers, 10 articles that had figures with error bars, but only 3 of them said what the error bars were. And these are all multi-author papers. So presumably, for this figure here, one of the authors decided we need to show error bars and worked out whether they should be little or whether they should be big and so on. But they were too naive or too much in a hurry or too forgetful because they forgot to say what the error bars were in the figure legends or in the materials and methods. And the other authors of these papers, so 7 out of 10 papers in Nature, also clearly haven't understood the paper on which they're an author, or they haven't read it, because they didn't say to the first author, oh, you forgot to say what the error bars were. And the same is true for the reviewers of these papers. And presumably each one got reviewed by at least 3 reviewers, but the reviewers were either incompetent or they didn't read the paper thoroughly because they didn't say, oh, look, the authors haven't said what the error bars were. And the same is true for the editors. So it's easy to conclude that 7 out of 10 papers appearing in Nature have not been carefully read by the editors, the peer reviewers, or even the authors of the papers. Right? So it's a bad state of affairs. It's bad because it means that most of these authors are not rigorous. It means that the journals are hopeless. It means the peer review process is very faulty. It does raise the question, why is it that these papers were accepted for publication in a journal such as Nature, even when they're so obviously flawed papers? And I'll let you think about that. So when I see this sort of thing, I get all steamed up and I write letters. So I wrote a letter to the editors of Nature saying, by not ensuring that all papers that have error bars describe what they are, Nature publishes material that cannot be properly assessed by its readers. And to give them credit, they published this letter and they improved their guidelines to authors and peer reviewers saying, oh, just check that the error bars, if they're shown, that they're described. So, and it's much less common for me to come across papers in Nature that show error bars but don't say what they are. Now, in some ways that's a good thing, things look a lot better, but in some ways it's probably a bad thing because it used to be easy to tell which 7 out of 10 papers in Nature to throw immediately in the trash, but now it's harder to tell the good ones from the bad ones. So that was Nature, but also a number of other journals, you know, I've written to and complained. So here's another journal. This is Proceedings to National Academy of Science, very prestigious journal. And if you're a member of the US Academy of Science, you can, or at least you used to be able to send about 5 or 6 papers a year to PNAS, and you got to choose the reviewers of the paper. That's if you're a member of the US Academy of Science. So this paper here, one of the authors is Bert Vogelstein, the most highly cited cancer researcher on planet Earth. And you can see he's a member of the US Academy of Science. He's contributed this paper. So I assume that he's also was the one who chose the reviewers of this paper. And in this paper here, you can see there's graphs and they've got these T-shaped things. The person up the back there with their hand on their chin, what are these T-shaped things called? And we'll go to the front where it's easy to see. What are the error bars in this case? Yeah, doesn't say. It doesn't say there or in the materials and methods. And yet, you know, these guys, presumably they know what error bars are. Maybe they haven't actually read the paper. And the same is probably true for the reviewers and, of course, the editors. So I wrote to Randy Sheckman, the editor-in-chief of PNAS, and I said, in this paper, figure 2, figure 5, supplementary figure S2 show graphs with error bars, but nowhere in the text, figure, legend, materials and methods is a description of them. So it's not possible to determine what they are. Please publish a correction so it's possible to interpret the data. Otherwise, readers may be left with the impression that papers appearing in PNAS have not been carefully read by the authors or the reviewers. And he was good enough to reply, "Dear David, you have been kind enough to point out this error on more than one occasion. And in spite of our efforts to tighten up instructions, authors continue to ignore us," right? So once again, you're going to be competing with people like this. You're starting out, you know, in your honours year, and you've got 1 million papers a year that are appearing that you've got to read, and yet a very large proportion of them, even from people as famous as Bert Vogelstein, are worthless, right? So part of this talk is to raise your skepticism, to make you ask questions about the papers you read, not believe something just because it comes from a famous author or a famous institution. So why show error bars at all? What's the point of showing error bars? I'll go for the tall guy with the white shirt in the middle there. Yeah, that one with glasses. Yep, and the mask. Yep. Why show error bars in a figure?
Because it's just sometimes, you know, the equipment is not accurate enough, so we need to have like a tolerance for the result.
I like to think there's two classes of reasons, two main reasons. And of course, you don't need to necessarily show error bars, and we'll discuss that a little bit too. But there are two broad categories of error bars. Error bars can summarise how the data are distributed. If you've got so many data points that it's just a cloud and confusing, you might show error bars as a summary statistic. Just like the mean is a summary statistic, it shows the mean of the samples, it gives you an idea where the mean of the whole population might be. By showing error bars of variation or range or standard deviation, it can show you how broadly or how narrowly the data are distributed. So it can summarise how the data are distributed. And these are called descriptive error bars because they describe and summarise the data. I'll ask the lady with the red top there with the key round her neck. How many broad classes of error bar are there? I'll give you a hint. It's somewhere between 1 and 3. 2, very good. And here's the other class. These are, so range, variation, standard deviation are descriptive error bars. And the other class of error bars are called inferential error bars. And they can help you draw inferences from the data, like they can indicate plausible values for the population mean, and they can be helpful when comparing sets of data, like if you're comparing the effects of a placebo on patients, the effects of a candidate drug. And they're called inferential error bars. Now, the most common inferential error bars are standard error of the mean and confidence intervals. And in clinical research, psychology, epidemiology, etc., where all of your evidence is statistical, if you're going to use error bars, use confidence intervals because they're the easiest to interpret. And many journal editors say you should, if you're showing error bars, they should be confidence intervals. In experimental biology, it's best not to use any error bars at all, just plot the data. If in a few circumstances there is a good reason for showing error bars, probably the ones to use are standard error of means because that's what's most common. And most of the time you want to show an inferential error bar because you're trying to draw an inference from your data rather than just saying what they are. But the main thing to do is if you're showing any error bars at all, you've got to describe what they are, ideally in the figure legend, so that the readers can easily understand what your data means. So let's go through an exercise and look at what sort of error bars we might use. Now imagine each blue dot there, and these are sort of binned, right? Normally there's measuring the length of a tail in a population of genetically identical blue-tailed mice, right? So these two mice had the shortest tails, this mouse had the longest tail. Most of the mice had tails somewhere in between. So it's about 36 mice randomly selected, and we've measured the lengths of their tails. Now, the guy right in the middle with the beard, with the black shirt, how many broad classes of error bar are there? 2. And which class would you use if you wanted to describe the data? No, which broad class? Starts with the letter D for describing the data. Descriptive, right, very good. Now, the easiest to understand descriptive error bars is the range, right? Just goes from the smallest data value to the largest data sample value, right? So everybody can understand the range. Now, it's descriptive, so if all of the mice had tails of roughly the same length, you'd have a very small range. With this particular genetically identical blue-tailed mice, you've got a very wide range, right? But if you just showed in your graph the range, you lose a lot of information. You're much better to plot the data. Certainly plot the data when you're looking at the data, and ideally plot all the data in your figures that you publish, or at least include all of the data in your supplementary material. Now this is the range for 36 mice. And let's imagine we randomly selected another 1 million mice from that same blue-tailed population. Would the range get bigger, smaller, or stay about the same? I'll choose the guy right in the back there with the laptop and the beard. Yep. It'd stay about the same. What do you think, the lady next to him? You've got a 30% chance. Bigger, smaller, stay the same? Stay the same. So we've got 2 saying stay the same. You just followed him, I can tell. Next one along, what do you reckon? Bigger, smaller, stay the same. We've increased the sample size to 1 million. It might increase, yes, all right. So we've got some controversy here. So who agrees that it stays about the same? Put your hands up. Who's too cowardly to... Who says it will get bigger? Who says it will get... Oh, look, that's good. Who says it gets smaller? Oh, it won't get smaller. Oh, it's very good to see bravery, even though it's completely wrong. The answer is it gets bigger, right? If you had 1 million mice, you'd have more and more outliers, right? You know, if you imagine if you started off with just 2 mice, that mouse and that mouse, then the range would be from there to there, right? That would be the range. And if you got 1 extra mouse, if it was out here or out here, then the range would get bigger, right? So if you had 1 million mice, you'd have some mice with practically no tail at all, and you'd have some Johnny C. Holmes of mice with really, really long tails. So that's why range is not often used as the descriptive statistic, because it's not robust. The range gets bigger and bigger and bigger. You get more and more outliers the more and more samples you look at. But it's a descriptive statistic. Now, a much more commonly used descriptive statistic is standard deviation. So this is the mean of the samples plus or minus 1 standard deviation. And standard deviation can be thought of as the average or typical difference between the mean of the samples and each individual sample. So about 2/3 of the samples are captured by the sample mean plus or minus 1 standard deviation. So this is again, descriptive statistics. If all the mice had tails of the same length, you'd have very narrow standard deviation. But in this population, it's quite wide standard deviations. Now, if we got another 1 million mice, would this interval get bigger, smaller, stay the same? Would the standard deviations get bigger, smaller, or stay the same? I'll choose the lady on the edge there. Yep. Stay the same. Stay the same. Next guy along, do you agree with her? Yeah, kind of. What about you? Get bigger. Good. Controversy. That's what I like to see. The guy just behind him a little bit. Stay the same. Next one. Get smaller. Oh, God, that's great. We've covered all the bases. So who agrees with her it gets smaller? Raise your hands. All right. Who says it stays about the same? And who says it gets bigger? Somebody said it before. You can't change mine. It stays about the same. It stays about the same because the standard deviation of the samples is always your best guess estimate of the standard deviation of the entire population. Just like the mean of the samples is always your best guess of the mean of the entire population. Now, of course, it will never be exactly the same. Like the mean of the samples might be a little bit bigger or a little bit smaller than the mean of the entire population. But it's your best guess, because that's all the data you've got. And the standard deviation of the samples is always your best estimate of the standard deviation of the entire population. So if you had a million mice, the standard deviation would get really, really, really close to the population standard deviation, but it might be a little bit bigger or it might be a little bit smaller than the standard deviation shown here. And that's why standard deviation is a robust statistic. Right? It's not like range. It doesn't get bigger and bigger and bigger the more samples you get. It just gets closer and closer to the population standard deviation. So that's why it's used very frequently as a descriptive statistic. Just like mean is often used as a descriptive statistic of the mean of the samples. So there's the mean of the samples and there's standard deviation. And obviously, you know the formula for mean. It's just the sum of the samples divided by the number of samples. And there's the formula for standard deviation. It's kind of like the average of the absolute difference between the samples and the mean of the samples. Now, if we selected another 36 mice, the mean of the samples might be here. Right? You know, if we select another 36 mice from the same population, it'd be very unlikely to have exactly the same sample mean. So that's another sample mean there. And if we select another 36 mice, it might have a mean there. And we select another 36 mice from the same population, it might have a mean there. Now, there's a distribution, a standard deviation of these means, and that's called the standard deviation of the means, also called the standard error of the means. And the expected standard error of the means is here's the formula. It's the standard error of the mean is the standard deviation divided by the square root of n, the number of samples, right? So the reason I've got it in a different colour from range and standard deviation is it's the other class of error bar that allows you to draw inferences. So it's an inferential error bar. And the sort of inference you can draw is the population mean is likely to be captured within this interval, the sample mean plus minus 1 standard deviation. It's probably going to be captured by that if you did the sampling 2/3 of the time. Now, if you had, sometimes people used to show the sample mean plus or minus 2 standard errors of the mean, and that's likely to capture the population mean roughly 95% of the time. So you can draw inferences about plausible locations of the population mean by using standard error of the mean. That's the sort of inference you can draw. Now, this is the sample mean plus or minus 1 standard error of the mean for 36 mice. Let's imagine we got 1 million mice. What would happen to this interval? Would it get bigger, smaller, or stay the same? The lady right in the corner there with the mask on. Yeah, turning your head in the ponytail. Bigger, smaller, stay the same. If you increase the sample size to 1 million, would the standard error of the mean get bigger, smaller, stay the same?
I would say smaller.
It would get smaller. Next one along, what do you reckon? Smaller. Next one? Smaller. Who agrees with them it gets smaller? Who says it stays the same? Who says it gets bigger? Oh, 100% correct. That's fantastic. And you can see the answers here because it's the standard deviation divided by the square root of n. So if n was 1 million, this will become 1,000 times smaller. It would give you a really, really accurate estimate of where the population mean is likely to lie. So the more data you have, the more accurate inferences you can draw. It won't change the standard deviation consistently one way or the other. It'll stay about the same, but it'll give you a more and more accurate estimate of where the population mean lies. And here's the other inferential error bar, confidence intervals, and formally this means that if you did this sampling from the same set of mice, 36 mice, if you did it 100 times, 95% of the time it would capture the population mean. Sometimes that sampling of 36 mice, that interval wouldn't capture the population mean. But so that's the 95% confidence intervals. Once again, it gives you a plausible interval within which you could expect to find the population mean. And you can see here it's roughly twice as wide as standard error of the mean. It's in fact, confidence intervals are related to standard error of the mean because it's just the standard error of the mean times the t statistic, and that gives the confidence intervals. Now, confidence intervals are often used, and again, it's an inferential statistic, are often used in clinical research if you're trying to look at the effect of a drug. So let's imagine we're trying to make a drug that increases the tail length in mice. And we've done double-blind trial where we've given some of the mice a placebo, let's say that's the blue ones, and we've given some of the mice this new drug to increase their tail length. And you can see just by inspection that these orange dots, the pattern is shifted slightly to the right. And if you look at the confidence intervals here, you can see that they're shifted and the sample mean is shifted slightly to the right. But the question is, well, is this a meaningful shift, or could you plausibly have a shift of this size just because of random selection of mice that you happen to select mice with slightly longer tails to give the drug to? So one way you can address that is by looking at the confidence intervals. Now, if the confidence intervals don't overlap at all, then you can say the differences are statistically significantly different. On the other hand, if the mean of one population is captured by the confidence intervals of the other population, then you can say the results are not statistically significantly different. And here, if there's a little bit of overlap, well, what you can do is you can talk to a proper statistician and they can enter your data into a computer, and then they can say, oh, look, the p-value is such and such, and it is statistically significant, it is the less than 0.05 level or whatever. But you shouldn't stop there, because you should look at the data. Now, this is just statistically significant, p is less than 0.05, but you've got to look at, well, how much difference is that going to make to each individual mouse? And the amount of increase in their tail length is relatively small. So your mice aren't going to be happy with this drug, even if the results are reproducible and statistically significant. And if we carried out this same experiment again, maybe we'd get results that haven't confirmed it. So if you do get a statistically significant difference, that's just a starting point. You've got to think, how do I confirm the results? What's the mechanism for that effect? What's the biological and medical significance of that effect? I mean, you can get a highly statistical significant effect and have the mice get shorter tails, but that's obviously not going to be a good drug. Any New Zealanders in the audience? None, not one. Good. Who's this guy? I'll give you a hint. He's a New Zealander. Do you know any physicists in the audience? Oh, hopeless. There's Ernest Rutherford, father of modern physics. He made some comments on statistics too. If your experiment needs statistics, you ought to have done a better experiment. And that's still true for a lot of experimental biology. You should design your experiments so that you get if you can, get black and white answers. You should triangulate your experiments. You should avoid just doing statistics. Now, if you're an epidemiologist, if you're doing big data sets and so on, if you're doing clinical research, then you need statistics. But if you're an experimental biologist, design your experiments so that you give black and white results that don't need statistics. Here are some bar graphs. These are showing mean plus or minus SEM. What are these T-shaped things called, lady at the front? Error bars, and in this case, standard error of the mean. And here's the data that's behind it. Now, you'd be much better to plot that data. Plot it when you're looking at your data, and plot it in the published paper, because you can see here you lose a lot of information when you just plot the means and the errors. In fact, these dots have exactly the same means and exactly the same standard error of the mean as these ones here, but if you plot it out, you'd see there's one dot up here. Maybe there was a mistake in copying down the result into your Excel spreadsheet, and that's a spurious result. But you get exactly the same errors and standard means and standard errors mean, and the same for this one. But if you plot it out, you can see these are populations with bimodal populations. You see lots more information if you plot the data, and the same is true for these ones. So obviously N is different for these two, but they give exactly the same means, exactly the same errors. So plot the data. Here is an XY graph where we're plotting the means of the x's, and you can see it only changes in the third decimal place. The means of the y's only change in the third decimal place. The standard deviations only change a tiny, tiny amount. Standard deviations of the y, even the correlation coefficient between x and y changes hardly at all. And yet if you plot the data, you can see a lot more information. Your brain is very good at picking up patterns by looking at plotted data. So you should always plot your data, and do a service to your readers and plot the data in the paper that's published, and if possible, publish the numerical results in a machine readable supplementary data set. So plot the data. So we're back to some practical examples. Here's a paper published in Nature. It's featuring Stan Korsmeyer, a good friend of Andreas Strasser, sitting up the back there. And in these graphs here are these little T-shaped things. The guy with the beard and the black shirt. Yeah. What are these T-shaped things called?
Error bars.
And what are the error bars in this case? I think he does say. Standard deviation, very good. But another thing you should always say in your figure legends is what N was, how many times they did the experiment. Now, in this thing, the results come from the mean and standard deviation from duplicate samples from 1 of 3 representative experiments. So N is 1, right? And if N is 1, you really shouldn't be doing any statistics, right? I mean, N is 2, that's kind of stretching it for standard deviation and so on, but N of 1, because these error bars reflect the differences in duplicate samples, right? Now, so these error bars just reflect the accuracy of their pipetting. They have nothing to do with whether white mice are different from the black mice. It's just saying here they were very careful pipetting, but here they're a bit sloppy at pipetting, right? But nothing to do with the hypothesis they're testing, because you can only draw hypotheses from independent data and not from duplicate or replicate data. And if they've done an experiment 3 times, as they claim, why didn't they show the results and plot the results of those 3 experiments rather than showing results from N equals 1? Here's another one. Again, I'm picking on Stan. The reason I'm picking on him is partly because he's dead, so he can't sue me, right? And here we have graphs. What are the T-shaped things called? The lady in the back there with the mask? Error bars. And what are the error bars in this case? Doesn't say. So we should throw the paper in the trash. It hasn't been carefully read by the authors or the reviewers or the editors. But we're going to keep looking at it because it's also mentioning this letter P. Now, I'll pick on this lady at the front there. What do these asterisks represent? And just yell it out. So say, what does that say?
Asterisk.
Yeah.
P smaller than 0.05.
Right, good. What word does P stand for?
P-value.
Yeah, it's P-value, but what word does it stand for? I'll pick on the next lady along. It stands for a word. There's a lot of words start with P. Probability, it is, good. Next one along. Probability of what? The probability of something is less than 0.05. Less than 5%, less than 1 in 20.
That the results are due to chance?
Not quite. It's the probability of getting a difference of this size or even bigger if you're sampling from a uniform population, right? If the null hypothesis was true. So it's not quite what you say. It's saying, what's the likelihood of getting a difference this big or bigger if you are sampling from the same population. Of course, you're not sampling from the same population, you're sampling from 2 different populations, so that's part of the controversy over p-values. Now, a lot of mathematicians say we should just get rid of p-values, and I tend to agree, but you'll come across them all the time, so it's good if you know a little bit about them, just so you know what to avoid and you know how to avoid them. So what is p? It's a measure of statistical significance. A better term would have been statistically noticeable, but it's not possible to wind back the clock. So it's just saying that something is statistically noticeable, right? There might be some signal above the background noise. That's all it's saying. It's the probability of observing a difference of this size or bigger when there really is no difference, meaning you're sampling from a single uniform population. It's the estimated false positive rate assuming the null hypothesis is true. So it's assuming that all of the mice are given a placebo and it's the chance of observing a difference of that size or even bigger. If p is greater than 0.05, it means there's no clear signal above the background noise. If a result is not statistically significant, you can't exclude the possibility that the differences you've seen are just due to chance. But you also can't conclude that there is no difference. The only conclusion you can make is that you can't make any conclusions. And statistical significance should not be confused with effect size or importance, right? So effect size is important, and things that are important, but statistical significance is not necessarily important at all. When n is less than 30, a better term for p is the laugh test. And when n is less than 10, a better term for P is the idiot's test, and it means if you do it, if you calculate p-values on your own data where n is less than 10, then that's what you are. And it's been confirmed. Right, so if it's statistically significant, consider, right, so only if you know, p is less than your value chosen, then you consider effect size, biological importance, past experience, underlying mechanisms, and how finding might be confirmed or refuted. And again, much better not to show p-values at all. If you want to show statistics, show confidence intervals. And if you're an experimental biologist, triangulate your evidence. So if you're wanting to prove the role of a protein, then and it's an enzyme, then do an enzyme assay. Then do an experiment where you're an inhibitor to that enzyme. Then do an experiment on cells in which you've knocked out the gene for that enzyme, right? So that evidence is not statistical, but it all builds together to give you a solid foundation, like Rutherford would have done, rather than just doing the same experiment again and again and again and again and again, and coming up with spurious results. So let's suppose we're looking at tail size in mice. Again, we've got blue-tailed mice, and here we've plotted, done the right thing, we've just plotted the size of the mice, plotted the data, but just for illustration, I've plotted the standard deviations and the standard error of the means. Now, these are wild-type mice, these are caudal knockout mice, and all of the knockout mice have very short tails, and all of the wild-type mice have tails that are roughly the same size. Now, anybody here used mice in experiments, or doing any genetic experiments on mice? All right. Do you think the caudal gene is having an effect on mice?
Yes.
Yes, I completely agree. Now, if you went to a statistician from Mars, they would say, oh, you can't make any conclusions where n is only 3. And I've just told you that you can't calculate p-values when you shouldn't calculate p-values when n is 3. But the thing is, we work in a Bayesian way. You've seen mice. You know that that's what their tail length is. Now, you know, if let's suppose you only had 2 mice, 2 knockout mice, 2 wild-type controls had 7-centimetre tails, 2 knockouts had tails 100 metres long. Right? Now, something's definitely going on, I believe, but if you talk to a statistician, they might say, oh, look, you haven't got enough data. Right? So you've got to think beyond, not all evidence is statistical. You've got to think beyond that. You've got to think about past experience, what potential mechanisms might be. And I would argue it would be unethical to just confirm this again and again and again, because we should use the minimum number of animals that give us a confidence in our results. But let's suppose we did with 10 mice, and that's the distribution there, and that's the distribution there. It's confirmed our results that there happens to be a difference in the mice. But let's suppose we knock out another gene, the Holman gene. Now we've got these mice up here, and these are the wild types down there. Now, who will I pick on that I haven't picked on yet? So I'll pick on the lady in red again, holding your gene with the key. Do you think the Holman gene is having an effect on tail length? All right, well, let's suppose we statistically looked at it and we found that the p-value is 0.017, right, less than 0.05. Can you conclude that there is a difference? Well, the hint was in the slides I showed you before, which this is an idiot's test, right? Because if I run the same simulation through the computer again, then I get p is 0.077, and there's no statistically significant difference. Now, you know, let's suppose I did a big study with large numbers and I got p is slightly less, or is equal to 0.05. If I repeated that same study, what's the most likely p-value I would get? Let's suppose I did it rigorously. I'll choose the guy in the middle there. Yeah. So I've done a big study, huge numbers of patients, and p equals 0.05. Let's suppose I replicate again, and I've done it thoroughly, rigorously, with exactly the same number, and what is the most likely p-value I would get?
Around the same, or just a little bit underneath?
The most likely one I get would be p equals 0.05 again. But there's a 50% chance p would be less than 0.05, 50% chance it'd be greater than 0.05. So there's a 50% chance if I repeated it, I wouldn't get a statistically significant result, right? So So once again here, especially with n's being very small, you can see that if you do the same things, the same emulation, the same built-in standard deviations, you can get significant results, non-significant results, much better to plot the data. And if you do see differences that are this small, you've got two options. Option one is to just get a whole lot more mice, and then these error bars will get a lot smaller, right? But I would argue that's not the ethical thing to do. You should think about, well, am I doing the right experiment? Maybe if I've got some reason for believing the Holman gene is involved in size of body parts, maybe I should be measuring whisker length, or maybe I should be looking at their white blood cell count. Maybe I should be genetically crossing the Cortland knockouts with the Holman knockouts to see if they act in the same genetic pathway. So if you see results that where the differences are very small and a small magnitude, I mean, think about the size of the effect, then redesign your experiment. Think like Rutherford. Don't just kill more and more mice. Now, as I said, we should get rid of p-values, and we should, and we definitely, all of us should never use p-values or even error bars for n as small as 3. We should plot the data. But I'm just going to tell you the rule of thumb of estimating p-values just by looking at standard error of the mean error bars. Now, here, imagine tail length in mice, and here are standard error of the mean error bars, and this rule of thumb is for standard error of the mean inferential error bars. Are these results statistically significantly different? If the bottom of one standard error of the mean error bar overlaps or just is very close to the top of the other standard error of the mean error bar, then the results cannot be statistically significantly different at the p is less than 0.05 level, right? So again, this is just a rule of thumb. You should plot the data, they should have plotted the data, but this is just something when you're looking at people's data and they tell you what the error bar, what the p-values are, just see whether that's consistent with what the data are telling you, right? So if standard error of the mean error bars overlap or are very close to each other, it cannot possibly be statistically significant at n is less than 0.05. And in this case, p is 0.2. Now imagine these are 3 different mice, and here there's a gap between the standard error of the mean error bars. So the rule of thumb is if you double standard error of the mean error bars, they'll double this one down to there, double this one up to there. If double SEMs, so a total of 4 SEMs, right, between that one and that one, if they don't overlap, then the result is statistically significant, at least at the p is less than 0.05 level. And in this case here, with only 3 mice of each type, double that one to there, double that one to there, it's very close. And I'll just ask this, do you think these results are statistically significant different or not?
Yes.
Yes? And why don't you have a punt at what p is?
Very close.
You're so good, so good. You get a gold star, right? 0.049, right? So these are just useful rules of thumb to think about when you're looking at data, and we're going to put them to the test now. Here's a paper published. It comes from Harvard Medical School, and it's by famous author Judy Lieberman, published in the Blood, and we're going to look at the difference between this bar, and I've blanked out the p-values because we're going to estimate them. Now, in this case here, they've said what n is. It's 3 independent experiments, and the error bars are standard deviations. So the rule of thumb I was telling you before was for standard error of the mean. Now, how do we estimate standard error of the means from standard deviation? We've got to divide standard deviations by something to get standard error of the mean. The guy with the striped T-shirt. What do I have to divide the standard deviations by to estimate the standard error of the mean?
The number of samples?
Close, close. It's the square root of the number of samples. And in this case, it's 3 experiments, so the square root of 3. Now, any medical students in the audience? Any wannabe medical students in the audience? Everybody's very shy, right? So I'll ask the lady there with the mask, what's the square root of 3? What would a medical student say? What's near enough? 2. 2 is the square root of 3. If you're a medical student, physicists would say something different, but near enough to 2, right? 1.7, you know, near enough to 2. Right, so the standard error of the mean error bar between this one, standard error of the mean would go from about there down to about there, and from here to go from there right to about there. So there'd be some overlap between down to there, So standard error of the mean would be down to about there. So there'd be overlap between this standard error of the mean and that standard error of the mean. So is this statistically significantly different from that? Nima, what do you think? No. The person at the back there with the beard and the purple t-shirt, is P greater than or less than 0.05? Yeah. What's that?
It's greater than.
Greater than 0.05. I completely agree. It's not significant. It's greater than 0.05. And yet at Harvard they say it's 0.01. 001. Now, I don't know how they get that, but I'm going to give them a second chance because there's another half of the graph over here. Now, let's compare this one to this one, right? N is 3. We'll estimate standard deviations go to there. Double two standard error of the mean would go down to there, right? And then divide by 2 and then double it. So now there's a gap between 2 standard error of the mean here and 2 standard error of the mean there. Is this statistically significantly different, just on face value? Is this statistically significantly different from that one over there? I'll choose that person there with the mask and the glasses. Yeah, you. Yes. All right. Do you want to take a, yeah, I agree. Do you want to take a punt at what P is? Yeah, I guess exactly the same. 0.002, something like that. Let's see what they calculate at Harvard Medical School. 0.2 by 10 to the minus 21, right? So I think they're out by about 10 to the 18, right? Or a billion fold. Now, I have no idea how they got to that result, but the onus is on the authors to make it comprehensible to the reviewers, but the readers, and I've got no idea. Maybe somebody leant on the 0 key when they were typing out their response. But the point is, just by looking at the graphs, looking at the p-values, you know you should throw this paper away. Here's another example. So now we're getting on to just one example of somebody doing the wrong thing. How's the time going? Well, I only got 15 minutes left. Right, so this is published in Nature, Massachusetts General Hospital, Harvard Medical School, Broad Institute and MIT. It's a preclinical cancer paper that Glenn Begley might have looked at this potential new drug called pipalongumine, and they're showing graphs and their error bars. They say what the error bars are, mean and standard deviation of 3 independent experiments. So they say what the error bars are, but these standard deviations are very small. And I'll just blow up that graph there. They're measuring tumour size in millimetres, right? So they're measuring length of tumours. And to me, that makes no sense at all, especially if they've dissected out the tumours, which they have. Why don't they measure tumour volume or weigh the tumours and say how many milligrams they weigh? Then you get an accurate thing. But they're measuring the length of the tumours. But are they measuring it from there to there or there to there or back to front? And the standard deviations are less than a millimetre, less than 1/10 of a millimetre, less than 100 microns in size in measuring length of tumours. And these tumours all look like different sizes, and yet they're the same controlled tumours down here, where the length of the tumour is less than 100 microns different, right? So these data just don't look plausible. And anybody who'd measured tumour size after dissecting them out, measure their length, they've got to be loons. And that means, and then look at all of these authors, right? Stuart Schreiber, Todd Golub, right? These are people who established the Broad Institute, and they've said length of tumours, and you know, I think they haven't read the paper, right? So another rule is if you write a paper and you're an author on it, you have to read it. But there is some evidence that they do very precise experiments. And another tip is when you're reading a paper, look at the supplementary material, because people are often not so careful in the supplementary material. But there is evidence here that they do very precise experiments. So here are some of the mice, and you can see these mice all have tumours. But this mouse has a tumour and it's also got a white patch on its back. And this mouse here also has a white patch on its back. And these tumours do look very similar. 3 of these tumours look similar. See, there's an ulceration in exactly the same position. They've done very careful experiments. You can see even the droppings in the floor of the cage are the same. You know, it's, they've done experiments so precise, it's almost as if they've just got one mouse and taken 4 pictures from 4 different angles. And that's why the variation is so small, and 3 pictures of one mouse down here. So it turned out that somebody wrote to the editors of Nature and said there's something funny going on, and they published a correction. And this is wording in their correction. We have been unable to verify, this is them telling the editors of Nature, without doubt that the image in supplementary figure 9b shows 4 different mice, right? We're unable to verify that we didn't fake our results. So does Nature ask them to then retract the paper? It says, no, 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 just publish a correction and show us a picture of another 4 mice. And here's their correction, a very extensive correction involving lots of things, different graphs, blah, 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 and there's some more mice. And so in their corrected graphs, now the error bars are plausible. Now they're measuring tumour volume, right? This makes a lot more sense. But some of the mice had tumours that must have been over 7 cubic centimetres in size. Now, a nude mouse weighs probably 20 grams, a little over 20 grams, so a third of its weight must have been tumour, right? Now, this breaches the animal welfare guidelines in Australia, in the US, in Europe, in fact, anywhere on the planet. But Nature has shown pictures of the mice, and here's pictures of the mice. These are enormous tumours. These dark stains here are ulcerations to the tumours. If anybody had mice like that in the Walter and Eliza Hall Institute's animal room, they would sack the animal technician, they would sack the PhD student, they would sack the honours student, they would sack the lab head. They would consider criminal prosecutions because this is a breach of animal welfare, right? And it shouldn't be permitted and it shouldn't be tolerated. And yet Nature publishes this by authors who've admitted they might have faked their results in the first place. So I wrote to the editors of Nature and got this reply saying, and so now this is a year after the paper was first published. This is to let you know in one of the upcoming editions of Nature we'll publish a second correction to this paper in which the authors state the experiments were not performed in compliance with the Institute's regulations. So they've admitted they might have faked their data. They've admitted they're not in compliance with the Institute's animal welfare regulations, and Nature still won't retract the paper. And so I asked them to publish an editorial saying how bad it was, and this is now another 3 years have passed, and I'm saying, how come you still haven't even published the second correction? And Philip Campbell, the editor-in-chief, saying, oh, it's very regrettable, it takes a long time, blah, blah, blah. 3 years have passed. But eventually, they did publish another correction, and that was together with an editorial, and in their editorial they wrote, cases such as this could provoke a justifiable backlash against animal research. All involved scientists, institutions, funders and journals must do more to ensure that regulations are strictly observed. And that's true, but Nature itself needs to do more because in this same edition of Nature, they just published another correction. They didn't retract the paper. So I was mentioning that if you're judging papers, you should look at the supplementary material. Don't print out the paper because it wastes paper, it wastes ink. Look at the images on a computer screen, try capturing images, changing the background, tilting the screen if it's a laptop. These are some microscope images. Now, can anybody see anything unusual there? Or nod your head if you'd like me to change the contrast. Can I see some head nodding? I can see head nodding up the back there. I'll change the contrast. Now, look down here at this image here. You can see some straight edges. Why are there straight edges in a microscope image? And if you look even carefully at the individual pixels, you'll see that splodge here is the same as the splodge here and also the same as the splodge there. Every little pixel in the purple box is the same as those ones, and then the yellow ones, the yellow ones, the red ones, the red ones, right? Now, these sorts of coincidences could just occur by chance, right? But the chance would be less than the chance of a million monkeys typing at random, typing over the life of the universe, the works of William Shakespeare. It would be way less than that. And yet here it is. So it could be coincidence, but that's very unlikely. It could be accidental, right? Somebody's just accidentally lent on the copy key and the paste key and they accidentally done. Or it could be deliberate misrepresentation of data, right? But you be the judge. Which do you think it is? So I wrote to my friend Philip, dear Philip, find 2 PDFs, analysis of supplementary figure 31B, and it looks like it's done by electronically duplicating parts of the images. Please look carefully at files, take actions necessary so readers in Nature can have confidence in the data it publishes. So that's 2016, and they investigating, their whole team of people are investigating, but still, we get to 2017, still nothing has been done. And yet they publish another editorial. Image doctoring must be halted. Researchers and journals do need to do more to counter inappropriately manipulated images. Journals need to do more. So I wrote to my friend Philip. Philip, I read the excellent editorial in today's Nature, image doctoring must be halted. Researchers and journals need to do more to counter inappropriately manipulated images in science. Prompted me to ask how the investigations are progressing. Now it's more than a year since of the manipulated images in this paper in Nature. Please give me an update. And then another year passes. Thank you for your email. I appreciate it takes a long time. Reassure you, we're going to do something very soon. And then finally, they retracted the paper. Here's the retraction notice for this Nature paper. Some of the authors are still standing by the retraction. That's 2018, and also there was Harvard had there was a federal misconduct probe into the lab at Harvard, and that's because the United States has an Office for Research Integrity that is a federal body that can look at research misconduct. Australia is one of the only countries in the world that doesn't have one, and as mentioned before, one of the things I'm lobbying for is that Australia establishes an Office of Research Integrity and joins the rest of the world, because we're being left further and further behind. So there was a misconduct probe, and then in August 2021, so August of last year, there were some penalties. He had to return, or Harvard had to return $1 million in funding, and Sam Lee, I think, had to pay out $250,000 worth of his grants. Right, so the lesson is, look at the data, look at the supplementary data, think about the error bars, right? So on a lighter note, here's a paper published in Cell. What are these T-shaped things called? I'm doing a little bit of revision. They're error bars. What are the error bars in this case? You'll do at the front, just yell out. Read what's underlined. Yeah, you.
Sorry. Bars represent the average plus minus means from 2 experiments.
Right, so what do you think is strange there?
1 plus or minus means.
Yeah. So the average plus or minus means, right? So the average is the mean, right? So I thought that was just a typo, but here it is in another figure, same edition of Cell. Look how many authors there are, including John Reed, one of the most highly cited researchers. He publishes an average 1 paper a week for the last 25 years. And, he was head of research at Roche, right? And yet and this other figure, different figure, bars represent average plus or minus means. So I thought, well, maybe they meant to say standard error of the, but they've just copied and pasted the thing and put it down there, and they've left it out. But it hasn't been carefully read by the authors, hasn't been carefully read by the editors or the reviewers. But that's what I used to think, but here's another paper from the same authors, error bars, showing averages plus or minus the means. Here's another paper, same authors, right? Bars represent average plus or minus the means, right? So there's clearly a problem with the way science is conducted. These kind of, I think UC San Diego or somewhere like that. So, legitimate, high reputation universities and high reputation journals, and yet this is the standard. So before you believe anything, think about it. Think about what's plausible. Think, is this consistent? Think how you might prove something or disprove something. If this is the project you're going to be working on for your honours, make sure that you've replicated the results that your project is going to be based on, even if it's done by your supervisor, right? Because some supervisors aren't as good as others. And if you do disprove them, then you can publish that. And if you supplement the results, at least you can sleep in bed at night, and so can they. So if you do see something that you've got questions about, I think you've got a duty and responsibility to say something about it. Now, there are mechanisms whereby you can anonymously raise issues at WEHI. It depends, is this something that's published? Is it something that's not published? Is it something from a colleague at WEHI or something from somebody in Canada? But I think you've got a duty to do something. You can speak up. You can look at WEHI's website for where you can report things. You can talk to a research integrity advisor. WEHI has a number of research integrity advisors. Melbourne Uni has a number of research integrity advisors. They can tell you, give you advice. You can write to the journal. You can write to the authors. You can write to officials at the host institution. You can publish something anonymously on PubPeer if it's a published paper. You can write to me. I have a collection. We've just looked at the very tippy tip of the collection. About anybody who wants to, have more examples or give me examples to increase my collection, I'd be very grateful. If it's funded by the NIH, you can contact the Office for Research Integrity. But of course, Australia doesn't have an Office for Research Integrity. And the other lesson is to protect yourself, and so that you can make discoveries that are patentable and you can become rich, maintain your lab book. That's the other clue, the other tip to protect yourself, as well as do research that can be reproduced and is of value. And with that, I'll stop there and try to answer some questions if there are any. You can clap now.
Thanks, Davo. Do we have any questions in here?
Yeah.
With the reproducibility of the data, what sort of parameters were around what constitutes a reproducible experiment, like when they test
That's a very broad question. So there is a reproducibility effort to reproduce papers in psychology and papers in cancer biology. I don't know what they did at Genentech. They were just trying to reproduce it enough to decide whether they invest hundreds of millions of dollars in developing the drug. So everybody's got different measures of what they consider to be a reproduction versus a replication. Some things you can't replicate because you obviously have different groups you're sampling from. But it's not so much the details, it's just the idea of doing something again to see if you get a similar result, just to, check your work.
We have one question from Slido from Anonymous. Are the problems you highlighted, poor statistics, even worse, fake results, getting worse, better, or the same as proportion of publications?
Impossible to know, right? Because most people who deliberately fake things, they don't want to be caught, so they don't, they don't fill out data sheets saying I've faked this many things on this many occasions and da 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 da. So it's impossible to know. There's more awareness of it, partly because of the efforts of Retraction Watch, that publicised retraction. So that's a great blog to look at, Retraction Watch. There's also many countries have now got Office of Research Integrity, and there's a World Conference on Research Integrity every year. So there's greater awareness, there are more retractions, there are more corrections. But how much, this is presumably still the tip of the iceberg. Now, how big the iceberg is underneath, nobody knows and nobody can know.
Any more questions?
Good, off the hook.
One more slider, sorry.
Yep.
Why p smaller than 0.05 for significance? Who came up with this? Is there reasoning behind it, or is it just arbitrary and something that has stood the test of time?
It's just arbitrary. It was probably Fisher, I think, that came up with p-values. And then there's big fights between Fisher and the p-values and Neumann and somebody or other else. I can't remember their names, but the statisticians are always arguing about things. The take-home message is show confidence intervals. Well, rule number one is plot your data. Rule number two is if you are going to show any sort of statistics or error bars, show confidence intervals in addition to plotting your data.
One more, sorry.
Yep.
With less than 10n, how do you call significance out of an interval of confidence instead of p-value?
Well, so where n is less than 10, you should plot the data, right? And not worry about statistics. You should triangulate. If you have numbers and you put them into an Excel spreadsheet or you put them into a statistical package, it will tell you a p-value and it will calculate the confidence intervals for you. So, sometimes people find that they do the right thing, they plot the data, they plot the 10 points, and then the reviewers of the paper say, oh, you've got to show error bars. Now, those reviewers are idiots, but you've got to do what they say, otherwise your paper won't be accepted. So what you should do is you should plot the error bars in addition to plotting all the data. And then you can show whichever error bars you like, as long as you say what they are. I would say the best ones to show are confidence intervals and leave it at that and hope that the reviewers will now accept the paper. All right.
Any more questions? Last chance.
Yeah, so anybody can write to me and tell me your tales of woe yeah. So thank you.